So another genetic pathway that has been identified to regulate lifespan in yeast is what's known as the mTOR pathway, or target of rapamycin. And these genes were identified using a drug screen with the immunosuppressant drug rapamycin and looking for any genes that actually responded to a long-term treatment with that immunosuppressant. So through that genetic screen, mTOR was identified. And what the mTOR gene encodes is the mTOR protein, which is a protein kinase. And kinases, as we know, phosphorylate things. And so mTOR's job in general is to act as a kinase in a signaling pathway, and it can phosphorylate several different cellular targets and actually promote protein synthesis as well as cell growth and cell survival and reproduction. So mTOR promotes or turns on protein synthesis, cell growth, and cell reproduction. And it does this in response to nutrients as well. And so we talked previously about how the SIR2 sort of pathway responds to environmental cues, including nutrition. And so mTOR responds to nutrients as well. And those nutrients will result in secretion of growth factors, which can bind to receptors in the cell membrane and activate what's known as a kinase cascade, and ultimately activate the mTOR protein kinase to promote these different processes, right? And so you can imagine that when there's a lot of nutrients around, activating protein synthesis, activating cell growth, and activating cell reproduction by phosphorylation makes a lot of sense because you have the nutrients available to you to start making proteins and to start making more cells. And however, what's interesting about this is that because mTOR promotes both the protein synthesis and resource-rich other processes like cell reproduction, it can actually decrease the lifespan of an individual. And repression of mTOR can actually increase lifespan. And so we're thinking here not necessarily about a whole organism in terms of like a human, but we're thinking about a unicellular yeast, right? And so every time that yeast cell divides, as we mentioned previously in the ERC lecture, it's one step closer to death. And so the more the cell is reproducing, or the more mTOR signaling there is, the shorter the ultimate lifespan. And that's why expression of mTOR actually decreases lifespan, because it forces the yeast cells into reproduction. And the more they reproduce, the shorter they ultimately live. And then when you repress or when you decrease mTOR gene expression, you can extend lifespan by preventing those cells from synthesizing proteins, from utilizing a lot of resources, as well as from reproducing. The same can be done at the protein level by using that rapamycin immunosuppressant drug to inhibit the protein's function as well. So you can either repress gene expression of mTOR to extend lifespan, or you can actually treat cells like yeast cells with this drug and stop the kinase from being able to phosphorylate, with ultimately that same lifespan extension phenotype. And you can actually see this borne out here in this experiment. So this is a lifespan comparison of wild-type yeast, which are this BY4741 strain, and a TOR1 or an mTOR deletion. You can see the delta strain here. And so this mTOR deletion strain in red is actually has a longer replicative lifespan in days than does the wild type. And that's because deleting that mTOR will actually stop the yeast from reproducing as well and extend lifespan. So whether you inhibit the mTOR pathway with rapamycin, whether you delete the mTOR gene, as you can see here, or if you just decrease mTOR expression, you can extend lifespan in all of these different ways. And so one thing that I do want to note is that both of these pathways, both SIR2 and mTOR, have been identified in other model organism systems as well as genetic regulators of lifespan. And so we're going to come back to both SIR2 and mTOR later when we talk about C. elegans as well as when we talk about mice.